Hello, everyone. It's my great honor that I have this opportunity to make presentation concerning of our original research. The title is Exploring hepatic metastasis of hepatocellular carcinoma based on methylation driver genes and establishing a prognostic model for hepatocellular carcinoma. And I come from the Department of Gastroenterology and Hepatology, the second affiliated hospital of Harvard Medical University, China. And I'm Professor, MD, and PhD. Here's my name is Shi Zhujin. Now, firstly, I will report my personal information. My name is Shi Zhujin, and the title is Professor, Chief of Physician, Doctor, Tutor, Postdoc, Cooperative Tutor. Position is Director of Part 1, Department of Gastroenterology and Hepatology, the second affiliated hospital for Harvard Medical University. Directions is a basic research on stem cell transplantation for digestive system disease. Also, as well as you know, I'm a clinical doctor and I also perform endoscopic surgery. It is minimally invasive strategy to treatment different type of gastro and hepatology diseases. And my degree is MD and PhD. Professional activities, member of the National Natural Science Foundation of China's first instance review expert pool, a reviewer of mechanisms such as stem cell research and survey, DDS and CDD, editorial board member of Journal Gastroenterology and Hepatology. Corresponding editors board member of Chinese Medical Association, HSA World, special reviewer of Journal of Practical Oncology. Main achievements. Until now on, I have published 44 essay paper. The highest impact factor is 17.5 to 1. The correlative impact factor was 209.635. The total number of citations was 1,249. H index is 19. I will from the following content to make the presentation. It is background, methods, results, and my conclusion. Firstly, I will introduce our research background. Primary liver cancer consists of two main categories: hepatocellular carcinoma, we call this HCC, and intrahepatic cholangiocarcinoma, ICC. Malignancies that originate the bile duct are ICCS, and those that originate in hepatocytes, which are the main parenchymal cells of the liver, are HCCS. More than 75% of primary liver cancer cases worldwide are characterized as HCC. As the seventh most common tumor, HCC is also the second main reason of cancer-related deaths. Studies have shown that mortality and morbidity rates were nearly equal in SCC patients. SCC is often diagnosed at a later stage and therefore has a higher mortality rate. Our research will form the following methods: from data processing, enrichment analysis, and methylation driver gene analysis. In fact, from data processing, where DNA methylation profile and RNA expression data were gathered from the TCGA and the GSE 11615 data sets, along with relevant clinical information. HCC with and without extrahepatic metastasis was classified according to the seventh edition of the ACGG, the version of 7 of ACGG. The development of tumors in stage 1, 2, and 3A and 3B is confined to the inside of the liver, while cancer tissues in stage 3C and 4 gradually and distantly invaded the liver. Second is enrichment of analysis. In this step is kind points to us in detail biological process, cellular component, and the molecular function together constitute the goal analysis. Goal analysis and the KEGG pathway enrichment analysis were performed for the aberrantly expressed genes and differentially methylated genes, respectively. Third, methylation driver genes. Gene with log FC is over 0.6 and p less than 0.05 were considered to the aberrantly expressed. Gene with log FC is over 0.1 and p less than 0.05 were regarded as aberrantly methylated. Genes with correlation over 0.30 and p less than 0.05 were considered as methylation driver genes. The fourth, prognostics model. Corpse regression analysis was conducted and a risk score model was built. A two-thirds of a truncation of the risk score was obtained to divide the samples into different risk groups. A prognostic model was established and a nomogram was drawn using C-index collaboration charts and ROC curves to evaluate the model. Then is immunohistochemistry examination. In detail, purified embedded ICC samples were obtained from the second affiliated hospital of Harbin Medical University in the Department of Surgery. With the consent and approval of the subjects and the second affiliated hospital of Harbin Medical University Ethical Committee. In detail, the ID is KY 20 and 23 and 49 samples from 3 HCC patients with extrahepatic metastasis and 3 HCC patients without extrahepatic metastasis of immunohistochemistry analysis. The sixth is immunoinfiltration. Ciprasol is a classical devolution method rooted in linear support vector regression, in which mRNA impressions level are used to quantitatively analyze the population of different immune cell subsets in a tumor tissue. The bar charts were constructed to reflect the difference in immune cell infiltration between different groups. In fact, studies have shown that 14.5% to 42% of HCC patients typically develop extrahepatic metastasis. HCC can metastasize outside the liver via direct spread, hematogenesis, dissemination, or lymphatic invasion. HCC patients mostly had intrahepatic stage 3 or IVA tumors when extrahepatic lesions metastasized, and the common metastasis site of HCC included lungs, lymph nodes, bone, adrenal glands, and peritoneum and omentum. Although the directions and the treatment of method of HCC have improved, the diagnosis of HCC patient typically occurs at the terminal stage. The survival time of patients has not significantly increased, and the 5-year survival rate remains less than 12%. By studying HCC patients with extrahepatic metastasis, research reported that 48.8% of patients who responded to treatment were alive after 1 year, 12.1% were alive after 2 years, and the survival rate of 1 year was 11.4% of patients with stable of prognostic diseases. With the continuous development of imaging techniques and an increasing number of methods can be used for detect sympathetic extrahepatic metastasis, including magnetic resonance imaging, computed tomography, ultrasound, and digital subtraction angiography. In fact, surgical treatment can improve the survival rate. However, only about 20% of patients are suitable for this option at the initial diagnosis. Spreading of the regional lymph node nodes and the distant metastasis are clearly contradictions for resection. Patients with limited hepatocyte, reversed or advanced tumors are not candidates for curative surgical treatment and can only receive palliative care. These patients exhibit a survival rate that is lower in comparison to patients who have been diagnosed with HCC at an early stage. DNA methylation is a considerable component of epigenetic modification, playing a crucial role in cell function, metabolism, and the regulation of carcinogenesis. Simmons et al. Found that lower expression of TMEM106A is significantly associated with hypermethylation of TMEM106A in HCC samples. In highly aggressive and metastatic HCC cell lines, the TMEM106A promoter is hypermethylated. However, the mRNA and protein levels of TMEM106A are reduced. Hamada et al. Collected normal and tumor tissues from patients with primary lung adenocarcinoma who underwent complete pneumococcal and reported that JNF132 is silenced in lung adenocarcinoma due to the DNA hypermethylation, and that DNA hypermethylation of this gene during normal to tumor tissue transition is significantly associated with vascular infiltration. Sun et al. Sequenced and analyzed paired DNA methylation and expression in lung cancer samples, calculated the correlation coeffective between the methylation changes of intersecting gene pairs and gene expression after screening, and identified 20 high methylation and 11 low methylation driver genes. Now I will report my results concerning our research. Definition CPGS and MRS. All 251 abnormally methylated CPGS, including 171 hypermethylated and 80 hypermethylated CPGS, were detected in the difference analysis between HCC samples from patients with and without extrahepatic metastasis. The CPGS were annotated as genes and 198 abbreviatedly methylated genes were identified, including 65 hypomethylated and 133 hypermethylated genes, compared with non-HCC metastatic tissues. 669 genes were overexpressed and 354 genes were downregulated in HCC metastatic tissues. In the enrichment analysis, for BP, differently methylated genes were mainly enriched in the regulation of transcription from the RNA polymerase II promoter and cell-cell signaling. For CC, differentially methylated genes were significantly enriched in extracellular exosomes, chromatin, and intracellular membrane-bound organelles. For MF, differentially methylated genes were enriched in metal ion binding and tau protein kinase activity. In KEGG analysis, differentially methylated genes were significantly enriched in vascular smooth muscle contraction, the releasing signal pathway, and the PI3K AKT signaling pathway. research status, and for BP, different expressed genes were involved in cell adhesion, chemical synaptic transmission, and response to extrahepatic stimuli. For CC, different nasally impressed gene in the cell surface and the apical plasma membrane. For MF, different nasally impressed genes were related to serine type and peptidase methylendopactase, and sulfotransferase activities. In KEGG analysis, brilliantly impressed genes were involved in the PPAR signaling pathway, glycine and serine metabolism, and other pathways. Methylation driver genes find it is 14 overlapping abrilliantly impressed and brilliantly methylated genes were observed. The correlation between the methylation and the impression levels of the gene were further calculated, and the curve over 0.30 and p less than 0.05 were identified at the cutoff levels. 8 methylation driver genes were identified. It is ANXA6, CRIP3, DHX58, DDP4, EF5A2, ETNK2, FOX L1, and PIPOX. Prognostic risk score. An related Cox analysis was conducted to identify for genes highly associated with extracellular extrahepatic metastasis of HCC p less than 0.05. Multivariate Cox regression analysis was performed to further refine the HCC risk score model, with the risk score being calculated using the formula risk score equal DHX58 multiple 0.308119276955271 at EF5A2 multiple 0.276.46679 542202. Using a cutoff level of 2/3 of the risk score, patients in the TCGA and the GSE 116174 sets were grouped into different risk groups, with a high risk group solving shorter OS. Prognostic risk score result confirmed the AUCS of the risk score model for 1, 3, and 5-year OS in the TCGA set were 0.62, 0.67, and 0.65 respectively. Moreover, in the GSE 116174 set at CC samples were further evaluated, and the AUCS at 1, 3, and 5-year were 0.62, 0.59, and 0.63. The gene impression and risk score distributions in the TCGA and the GEO database showed as the following. It is a higher risk and a lower risk. We can increasing in patients, increasing risk score is separately, and died and alive use different color around the sample. It is a red color is standing for dead, and green color, blue color standing for it is alive, and we can face increasing score. Survival time, it is following different years, and the patient number as following go on, we can calculate the detail as separately. Prognostic model. The clinical pathological factors obtained from the TCGA and GSE 116174 data sets are presented. Cox analysis showed that the prognostic risk score and the pathological states exhibit strong association with OS. In fact, it's a p less than 0.05. It is we can carefully check the prognostic model as followed. In different database, overall, we will check the and the stage different. We can check the overall prognostic model. A Cox regression model, including risk score and the pathologic stage, was built to predict HCC prognosis, and a nomogram was drawn as followed. We can see his points and the risk score and the different stage and the total points from stage 1, stage 2, and stage 3A and stage 3B. We can calculate the prognostic risk score. C-index calibration and ROC curves were plotted to evaluate the prognostic model. The C-index in the TCGA set was 0.668, and that in the GEO set was 0.608. The calibration curves of the TCGA and the GEO sets close to the ideal curves. In fact, it's 45%, and the measured values agreed reasonably well with the predicted values. The 1, 3, and 5-year AUCS were 0.72 and 0.74 and 0.69 in the TCGA set, and 0.6 and 6 and 0.62 and 0.68 in the GEO set. And finalized, we use immunohistochemistry to validation our research by experimental research. In fact, we can subgroup metastasis and non-metastasis to compare with two types of antibodies impression level, DX58 and EIF5A2 as separately, to identify the impression of the Chi-Haba genes, DX58 and EIF5A2 pathological specimen from HCC patients were subject to IHC staining. The AOD of each image was then measured using the ImageJ software. The cumulative optical density was divided by the total area to obtain the AOD. Immunohistochemical demonstrate that the impression of level EIF5A2 in extrahepatic metastatic specimen was found to be higher compared to in HCC non-liver metastatic specimens, and the expression of DX58 in extrahepatic metastatic specimens was lower than that in HCC non-hepatic metastatic specimens. To further evaluate these, HCC samples from the TCGA cohort were immunoinfiltration analyzed, revealing that the abundance of M0 macrophages was high in the high-risk group, p less than 0.05, and was positively related to EIF5A2 impression, p less than 0.05. Additionally, Figure 6L demonstrates that M0 macrophage abundance and risk score were positively correlated in HCC samples. This is consistent with the previous study by Weijie Zhou et al. Here is immunoinfiltrating samples. We can judgment from this image to show vividly, especially to check the macrophage M0, macrophage M1, and macrophage M2. Here is typically result concerning of immunoinfiltration. And how, in briefly, we can use this mechanism to depict our research and explain the detailed mechanism by the image showed as followed. We can find in combination with our own studies, it can be speculated that LGP2 inspected DX58 as a positive regulator of IIG1 participates in PI3K AKT activation via the FTX MER545 axis of NCRN non-coding RNA by targeting IIG1, leading to HCC extrahepatic metastasis. And our study proposed a hypothesis that mechanism by which EIF5A2 promotes HCC extrahepatic metastasis. EIF5A2 activities the activity of RH0 GPT pace to stimulate actin cytoskeletal rearrangement and further induces the progression of EMT through activation of the ROS pathway, thereby promoting HCC extrahepatic metastasis. And now we can see this extrahepatic metastasis of HCC from adrenal gland, lungs, omentum, and lymph node extracellular. It is a long distance metastasis. In non-extrahepatics of HCC, or also we are also to establish a model to evaluate this PI3K and AKT. Conclusion: methylation driver genes. 8 methylation driver genes were identified as followed. Just now I have shown you NSA6, CIIP3, DS58, and DPP4, EF5A2, ETNK2, FOXL1, and piperoxen. Risk score is a Cox regression analysis was performed to refine the HCC risk score model. We establish the risk score and immunohistochemical. Immunohistochemical verified that the expression level of EIF5A2 in extrahepatic metastatic specimens was found to be higher compared to HCC non-liver metastatic specimen. And the expression of DX58 in extrahepatic metastatic specimens were lower than that in HCC non-hepatic metastatic specimens. At the same time, we perform immunoinfiltration. Immunoinfiltration analysis revealed that the abundance of M0 macrophages was high in the high-risk group and was positively related to EIF5A2 impression. We hypothesized that EIF5A2 impression works synergistically with M0 macrophage abundance to promote HCC progression and influence the prognosis of HCC patients. In detail, we explain the mechanism as follows. We speculated that LGP2 as a positive regulator of RIG1 participates in PI3K AKT activation, where the FTX MER545 axis of non-coding RNA by targeting RIG1, leading to HCC extrahepatic metastasis. And we proposed a hypothesized mechanisms by which EIF5A2 promotes HCC extrahepatic metastasis. EIF5A2 activates the activity of RH0 GT pace to stimulate actin cytoskeletal rearrangement and further induces the progression of EMT through activation of the ROS pathway, thereby promoting HCC extrahepatic metastasis. Here's a conclusion and our mechanism Totally, we report our research in briefly to let all of our friends to understand this research, and you know it is a great opportunity. It is my honor and thanks of you to take your long time to listening for my report, and in exploring is extrahepatic metastasis. We find these methylation driver genes in our research. Again, thanks for your patience and thanks for your cooperation.